On Tuesday, I introduced DNA hairpin as an interesting structure that we can consider as a model system to study two-state dynamics between closed and open. And the population difference between the two, ratio between the two can be described with a single number that is a free energy difference between the two states. I also introduced the transition state theory that basically tells you how you can think about the rate of transition between two states, from one state to another, based on the height of the energy barrier, or delta G dagger. And one way to learn more about the nature of the transition state was to use a force that you can apply to the molecule to bias the reaction. And by measuring how much the bias the force can give you, you can determine the distance to the transition state, and that can limit the possible models arising. So one example that I can just go over is that let's imagine a DNA hairpin, you're applying the force. And then let's imagine that you have a fluorescence molecule that is attached here. And then a molecule that you can call Q for quencher. So if you measure like a signal from this molecule, then this is fluorescence intensity and time. And in this closed state, you excite the fluorescent molecule using a laser, but it is quenched by the adjacent quencher. So your fluorescence signal will be very, very low. Okay? And then it opens up. And these two guys are now widely separated. Then and no longer quenched, this will, emit light, and then this signal will suddenly go up. Okay. And then as it comes back, then it comes down and so on. So you can imagine seeing two-step fluctuations. Now, if you measure this at different forces, now let's imagine performing several different experiments as a function of time, but as you increase the force, so at low forces, then you prefer this closed state. So your fluorescence will remain very low, and occasionally maybe spike up, and so on. And if you go to very high force, then you are almost always spending your time in this open state. Your signal will be like this, and somewhere in the middle you'll see about half and half between the two states, and here it'll look more like this, and you get the idea. Okay? So you can actually measure these transitions from a single molecule as a function of force, and then you can actually measure the rate of transition between two different states, and then, for example, rate of going from closed state to the open state as a function of force. If you plot the log of that value, as you increase the force, this rate will increase, and it will increase exponentially with force. So in the log scale, you get a line like that. And if you measure the rate of going from open state to the closed state as a function of force, it will decrease. And the idea is that by looking at the slope, here you can deduce the distance to the transition state, because the slope slope is basically equal to delta x to the transition state over kt. Okay. So I think about 2 weeks, or 2 or 3 weeks from now, Professor Jan Schemner will give a guest lecture to introduce you to the concept of optical tweezers, optical trap, that can be used to actually apply force in a controlled manner. So I will not actually tell you how these measurements are actually done. Questions? I got some Halloween candies for you. Neil? Yeah? There. All right, so that was a really quick review. So today, I want to talk about like 3 different topics. You can find the DNA flexibility in the lecture notes, so we'll get to that. But I want to first actually talk about DNA origami and Encode. Do you know what origami means? So it's a Japanese word. Ori means fold, and kami means paper. Actually, I'm not so sure. It can be the other way around. My father-in-law told me that, and I forgot which one is which, but it means a paper folding. Okay? But you can actually fold a nicely well-defined structure using DNA. And I have an interesting TED Talk that I want to share a part of with you. And also interesting publication. Video? Okay. By the way, Jingyi Fei is sitting here. She's going to be giving you the lecture on Tuesday. And she wants to see what I do, and then she wants to mimic my exact mannerism next week. So you'll see how she does this next time. Okay, so this paper was published in 2006, so that's already 7 years ago. And title is Folding DNA to Create Nanoscale Shapes and Patterns. And it was published by a single person, Paul Rothenmund from Caltech. And this is something that I always wanted to do, to publish a single author paper, so I can say, here I describe a single. Right, so anyway, in a nutshell, what he did was to develop a method to, make nanoscale structures using DNA. Okay, and so these are 2 subpanels from the paper. You know what that is? Star. And what is this? Wrong. In the paper, you cannot find that expression. Okay. Okay. He wants to show that DNA origami need not be topological disk, meaning that you can also have holes, right? A 3-hole disk was the design. So official name for this structure is a disk with 3 holes. Okay. Okay, let me show you his talk. Okay. Nice. Isn't it interesting? It's nice presentation. And also, it kind of connects what we discussed so far in the class too. Did I lose the internet?
Shouldn't.
Have you watched a TED Talk before? I see a lot of interesting things. It's really quite...
So people argue vigorously about the definition of life. They ask if it should have reproduction in it, or metabolism, or evolution. And I don't know the answer to that, so I'm not going to tell you. I will say that life involves computation. So this is a computer program. Booted up in a cell, the program would execute, and it could result in this person. Or with a small change, it could result in this person, or another small change, this person, or with a larger change, this dog, or this tree, or this whale. So now if you take this metaphor as genome as program seriously, you have to consider that Chris Anderson is a computer fabricated artifact, as is Jim Watson, Craig Venter, as are all of us. And in convincing yourself that this metaphor is true, there are lots of similarities between genetic programs and computer programs that could help convince you. But one to me that's most compelling is a peculiar sensitivity to small changes that can make large changes in biological development. The output, a small mutation can take a 2-winged fly and make it a 4-winged fly. Or it could take a fly and put legs where its antenna should be, or if you're familiar with The Princess Bride, it can create a 6-fingered man. Now, a hallmark of computer programs is just this kind of sensitivity to small changes. If your bank account's $1 and you flip a single bit, you can end up with $1,000. So these small changes are things that I think that they indicate to us that a complicated computation in development is underlying these amplified large changes. So now, all of this indicates that there are molecular programs underlying biology, and it shows the power of molecular programs, biology does, and what I want to do is write molecular programs potentially to build technology. And there are a lot of people doing this, a lot of psychologists doing this, like Craig Venter, and they concentrate on using cells. They're cell-oriented. So my friends, molecular programmers, and I have a sort of biomolecule-centric approach. We're interested in using DNA, RNA, and protein and building new languages for building things from the bottom up, using biomolecules potentially having nothing to do with biology. So these are all the machines in a cell. There's a camera, there's the solar panels of the cell, some switches that turn your genes on and off, the girders of the cell, motors that move your muscles. My little group of molecular programmers are trying to refashion all of these parts from DNA. We're not DNA zealots, but DNA is the cheapest, easiest to understand, and easy to program material to do this. And as other things become easier to use, maybe protein will work with those. If we succeed, what will molecular programming look like? You're going to sit in front of your computer, you're going to design something like a cell phone, and in a high-level language, you'll describe that cell phone. Then you're going to have a compiler that's going to take that description, and it's going to turn it into actual molecules that can be sent to a synthesizer, and that synthesizer will pack those molecules into a seed. And what happens if you water and feed that seed appropriately is that it'll do a developmental computation, a molecular computation, and it'll build an electronic computer. And if I haven't revealed my prejudices already, I think that life has been about molecular computers, building electrochemical computers, building electronic computers, which together with electrochemical computers will build new molecular computers, which will build new electronic computers, and so forth. And if you buy all of this and you think life is about computation as I do, then you look at big questions through the eyes of a computer scientist. So one big question is, how does baby know when to stop growing? And for molecular programming, the question is, how does your cell phone know when to stop growing? Or how does a computer program know when to stop running? Or more to the point, how do you know if a program will ever stop? There are other questions like this too. One of them is Craig Venter's question. Turns out I think he's actually a computer scientist. He asked, how big is the minimal genome that will give me a functioning microorganism? How few genes can I use? This is exactly analogous to the question, what's the smallest program I can write that'll act exactly like Microsoft Word? And just as he's writing, bacteria that will be smaller, he's writing genomes that'll work, we could write smaller programs that would do what Microsoft Word does. But for molecular programming, our question is, how many molecules do we need to put in that seed to get a cell phone? What's the smallest number we can get away with? Now, these are big questions in computer science. These are all complexity questions. And computer science tells us these are very hard questions. Almost many of them are impossible. But for some tasks, we can start to answer them. So I'm going to start asking those questions for the DNA structures I'm going to talk about next. So this is normal DNA. What you think of it as normal DNA. It's double-stranded. It's a double helix. It has the A's, T's, C's, and G's that pair to hold the strands together. And I'm going to draw it like this sometimes, just so I don't scare you. We want to look at individual strands and not think about
5 prime, 3 prime.
It comes single-stranded. So we can take the blue strand in one tube and make an orange strand in the other tube, and they're floppy when they're single-stranded. You mix them together, and they make a rigid double helix. Now, for the last 25 years, Ned Seaman and a bunch of his descendants have worked very hard to make beautiful three-dimensional structures using this kind of reaction of DNA strands coming together, but a lot of their approaches, though elegant, take a long time. They can take a couple of years, or it can be difficult to design. So I came up with a new method a couple of years ago I called DNA Origami that's so easy you could do it at home in your kitchen and design the stuff on a laptop. But to do it, you need a long single strand of DNA, which is technically very difficult to get. So you can go to a natural source. You can look in this computer-fabricated artifact, and he's got a double-stranded genome that's no good. You look at his intestines, there are billions of bacteria. They're no good either, double-strand again. But inside them, they're infected with a virus that has a nice, long, single-stranded genome that we can fold like a piece of paper. And here's how we do it. This is part of that genome. We add a bunch of synthetic DNAs that I call staples. Each one has a left half that binds the long strand in one place, and a right half that binds it in a different place, and brings the long strand together like this. The net action of many of these on that long strand is to fold it into something like a rectangle. Now, we can't actually take a movie of this process, but Sean Douglas at Harvard has made a nice visualization for us that begins with a long strand and has some short strands in it. And what happens is that we mix these strands together, we heat them up, we've got a little bit of salt, heat them up to almost boiling and cool them down. And as we cool them down, the short strands bind the long strands and start to form structure. And you can see a little bit of double helix forming there. When you look at DNA Origami, you can see that what it really is, even though you think it's complicated, is a bunch of double helices that are parallel to each other. And they're held together by places where short strands go along one helix and then jump to another one. So there's a strand that goes like this, goes along one helix and binds and jumps to another helix and comes back that holds the long strand like this. Now, to show that we could make any shape or pattern we wanted, I tried to make this shape. I wanted to fold DNA into something that goes up over the eye, down the nose, up the nose, around the forehead, back down, and end in a little loop like this. And so I thought if this could work, anything could work. So I had a computer program design the short staples to do this. I ordered them, they came by FedEx. I mixed them up, heated them, cooled them down, and I got 50 billion little smiley faces floating around in a single drop of water. And each one of these is just 1,000th, if they're 1,000th the width of a human hair, okay? So they're all floating around in solution. And to look at them, you have to get them on a surface where they stick. So you pour them out onto a surface, and they start to stick to that surface, and we take a picture using an atomic force microscope. We've got a needle, like a record needle, that goes back and forth over the surface, bumps up and down, and feels the height of the first surface. It feels the DNA Origami. There's the atomic force microscope working. And you can see that the landing's a little rough when you zoom in. They've got, they have weak jaws that flip over their head, and some of their noses get punched out, but it's pretty good. You can zoom in and even see the actual little loop, his little nano goatee. Now, what's great about this is anybody can do this. And so I got this in the mail about a year after I did this, unsolicited. Anyone know what this is? What is it? It's China, right? So what happened is a graduate student in China, Lulu Xian, did a great job. She wrote all her own software to design and build this DNA Origami, a beautiful rendition of China, which even has Taiwan, and you can see that it's sort of on the world's shortest leash, right? So this works really well. And you can make patterns as well as shapes, okay? And you can make a map of the Americas and spell DNA with DNA. And what's really neat about it, well, actually this all looks like nano artwork, but it turns out that nano artwork is just what you need to make nano circuits. So you can put circuit components on the staples, like a light bulb and a light switch, let the thing assemble, and you'll get some kind of a circuit. And then you can maybe wash the DNA away and have the circuit left over. So this is what some colleagues of mine at Caltech did. They took a DNA Origami, organized some carbon nanotubes, made a little switch, you see here, wired it up, tested it and showed that it is indeed a switch. Now, this is just a single switch, and you need 500 million for a computer, so we have a long way to go, but this is very promising because the origami can organize parts just 1/10 the size of those in a normal computer, so it's very promising for making small computers. Now, I want to get back to that compiler. The DNA Origami is a proof that that compiler actually works. So you start with something in the computer, you get a high-level description of the computer program, a high-level description of the origami. You can compile it to molecules, send it to a synthesizer, and it actually works. And it turns out that a company has made a nice program that's much better than my code, which is kind of ugly, and will allow us to do this in a nice visual computer-aided design way. So now you can say, all right, why isn't DNA Origami the end of the story? You have your molecular compiler, you can do whatever you want. The fact is that it does not scale. So if you want to...
Okay, so let me stop here. You can continue to watch TED.com. So the origami actually has a long history. The field was first studied by Ned Seaman at NYU, I think it was 30 years ago. And he was a structural biologist. He was using X-ray diffraction to solve the structure of biomolecules. But when he was an assistant professor, he couldn't crystallize any of the proteins he wanted to crystallize. And then he realized that, well, if there's no crystal, there's no structure, and there's no structure, there's no structural biologist. Basically, he would not get tenure. So his idea was to, instead of trying to convince proteins to self-assemble into a structure, he wanted to make DNA, artificial DNA lattice in 3D. And then attach a protein to the each lattice point, and then thereby solve the structure of the protein that way. That's why he began to do this, and he never actually solved any structure of any protein, but he really started a really exciting field. The Rothman paper is a really famous one from 2006. This talk was given in 2008, and already at this point, there's a nice video from Harvard University that was done by the group of William Shih, and they already had a 3D version of the origami working, but they couldn't publish for a couple of years because they didn't know how to actually take images of a 3D and then show that indeed you made a structure. So in the end, they had to do electron microscopy in 3D, cryo-EM, and so on to get that. So it's really a really exciting field. Can be used for replacing some of the DNA tethers for single molecule biophysics experiments, and a lot of cool things. So just wanted to introduce this to you. Questions?
How do you control the pattern?
How do you control the pattern?
Yeah.
Well, so the idea is that you have one circular DNA, and that you want to fold it into the shape that you want. So as long as you can fold it into a shape that has the same area, you can design these stapling DNAs to put them together. That's what the animation showed. And I think you can do it. It's gonna cost you probably about $500 to buy all of the stapling oligos. And the 2D was relatively easy to do, but the 3D origami took some years for the group in Harvard. And I talked to a German postdoc who used to be there. He's in Munich, and they couldn't really get it to work because in 3D, how do you make sure that all the right structures are formed in the right order? So in the end, what he had to do was to just this assembly process really slowly. So it took them over a week, right, to mix all of the DNA molecules, and then you go to high temperature and then slowly cool down over a 1 week period, and eventually they were able to get some of the molecules. But then actually the same guy early this year published a paper in Science showing that now you can do this in just a couple of hours, in half an hour. The idea is that when you go to high temperature and then you gradually cool it down, actual assembly occurs at a well-defined temperature determined by the structure itself. So you don't even have to start from high temperature and go down slowly. As long as you know at what temperature that occurs, just cool down very quickly there, and then you do the slow step around that sharp transition temperature. So it's just going to be even easier to do. Okay. Questions?
So they design the sequence and they mix a long strand with a lot of short pieces.
Yes.
And then code it down.
Yes.
And they will bind at some specific positions.
That's right.
That makes the pen.
Yes. So one long DNA is about 7 bases long, 7,000 bases long. That's actually virus DNA. This virus that infects E. coli, its genome is single-stranded, and about 7,000 nucleotides long. And then you can you can buy in bulk, cheap, and then you just buy smaller stitching DNA molecules to assemble this. Any other question? Okay. So Encode is a project that was funded by NIH for I think several years, and effort by many different laboratories. The idea is that DNA sequence alone does not determine the cell type or tissue type. There are other things that are different between cells, or different tissues, or different stages of disease and so on. So different labs actually collected data of all different kinds, not DNA, but modification of DNA, modification of the proteins bound to the DNA, or how many RNA molecules of different kinds are made in certain type and so on, so that you can form a central database people can look up. Is that fair, Jingyi? And non-coding regions. Non-coding regions too. So what does Encode stand for?
Encyclopedia of DNA.
Encyclopedia of. Okay. Oh, so we'll show a video. Okay, that's just. So this is a shorter one.
Like Gregor, you are built.
Produced with exclusive support from Illumina.
This is the story of you. Billions of years of evolution, and here you are, watching this guy. He is Gregor Mendel, an Austrian monk and scientist. Hello, Gregor. In the middle of the 1800s, he's going about his monkey business breeding pea plants, and discovers that certain traits are inherited as units, that his pea flowers are either purple or white, never a blend of the two, never purple-ish. We call these units of inheritance genes, and for this discovery, Mendel is known as the father of genetics, which, since monks aren't allowed to father much of anything, is both naughty and excellent. Like Gregor, you are built from around 50 trillion cells. Like him and his pea plants, your cells contain a set of instructions that you pass on to your children, your genome. It's all made of DNA, which carries a code built from an alphabet of just 4 letters, A, T, C, and G. But there's 3 billion of them. Gosh, that's awfully complicated. Since Mendel's staggering progress has been made deciphering genomes, here are some other 50 trillion cell folk, Watson, Crick, and Franklin, discovering the DNA double helix. And this lot, figuring out how the code is translated, how and when it's active, and what happens when it goes wrong. Genomes certainly are tricky. The next chapter in your story is the Human Genome Project. The entire 3 billion letter code that makes an average person was published at the start of the 21st century to great, but not literal, fanfare. Presidents heralded the end of disease, because presidents like to do that sort of thing. Unfortunately for presidents, it's not a purple and white issue. You are more complex than peas. Genetics is not just about the code, it's about how that code is interpreted, how it makes different cells and different people. To figure it out, someone has to look for differences between people. The story of you, and you, and you, and you, and a lot more yous to see how those variations combine and how they relate to disease. That's what this mob do. Compare and track genes, highlighting tiny bits in the story of you. They're also inventing faster ways to read genomes, resulting in explosions of data. Yeah, there we go. 10 years on from the Human Genome Project, the next colossal chapter in your story is the Encyclopedia of DNA Elements, or Encode. Take a bow. Encode is a monumental step towards piecing together how you and me and everyone work. It's a map that reveals the whole genome is a jungle buzzing with activity. Even the parts we used to think of as junk. Not just the genes, but instructions telling them what to do and when to do it. People and diseases are complex because our genomes are complex. Encode hugely advances our understanding of who you are, where you come from, and how genetic disorders such as cancers or heart disease happen so that one day they'll be fixable. So, here you are with your 50 trillion cells and your complicated code that makes you utterly unique. How your story begins and unfolds is something humankind is only just beginning to fathom. And that story, my friend, is a gloriously colourful thing.
So they put a lot of money into this project, probably several $100 million. Yeah. And
Produced with exclusive support from Illumina.
okay, this is a company that pay for this movie, not pay for the project. Did I mention Illumina before? Yeah. It's a company that dominates, dominating the sequencing market. So they actually sell most of the DNA sequencing machines in the world now. And the market value is about $10 billion. It all started from a startup company by two professors, young professors in Cambridge, London. The technology became actually a $10 billion company. So Encode is really, again, about how the gene expression is regulated. Again, it's not the gene itself, but when and where these genes are expressed that determines the fate of a cell, different tissues and disease state, normal state, and so on. So regulation is very important. The second half of my lectures, this semester will be on more about regulation and using several classic papers in systems biology and synthetic biology as some examples. Okay. All right. So then I want to stop the show and then get serious again. Okay. Questions? Oh, yeah, I do have the show. Okay. All right. Yes?
How do they study how the genes are expressed? So with the genome project, you mass sequence them. What is the tool that people use in Encode?
So Encode is largely RNA molecules. So you know that DNA is then converted to RNA and then to proteins. And actually, real full mapping would require protein mapping. That's quite a bit more difficult. You have to use other technologies such as mass spectroscopy, and you can do now the genome-wide studies of proteins, but it's still very expensive and difficult. So I think most of the thing is to measure the level of RNA molecules, okay, and use that RNA level as a proxy of protein expression. Questions? Yes?
So if I invent a method to quickly sequence proteins, Nobel Prize?
Yes.
Cool.
No, I mean, if you...
Right on that.
Yes, Nobel Prize, definitely.
Thanks.
Well, I can tell you several other things that you can win the Nobel Prize for.
Yeah.
Bring some candies next time. All right, so I want to talk about DNA flexibility. And here's my model of DNA. Actually, it got entangled. So I mean, okay. I asked my secretary to assemble a paperclip chain of the length 25 units. Okay. No, it's not 25. Actually, maybe did I lose it? 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay, only 20. Okay, I lost 5 somehow. All right, so if I look at this DNA, L is the length of the fully stretched chain length, and it is n times L. Okay, and it's the number of units I have. In this case, it's 20, and then it'll be about 1 inch. And so if I put this, I don't know. Okay, so I start with this, but then in the cell or in solution, there's a lot of thermal fluctuations, and this is small. The thermal noise dominates. So basically, if I let it go, then it'll come down in a random shape. And then if every time I do it, I get a different shape. But it's almost never going to be fully stretched, right? Because that has a very low entropy. It's not very preferred, right? So the question is, on average, when you do it many times, can you say anything about the average size of this chain? So that is the question. Okay? So let's draw the chain. So I have these vectors of length L, and this one, I guess you can call this L times a unit vector in this direction, right? And I have this all over, and so what number can I actually determine average over many different configurations? Where you can say, all right, so what about this vector connecting the beginning and the end point. So this is, let's say, so then that distance will be adding all of these vectors together. Okay, and then if I want to now ask myself, okay, what is the average size? Then I have the average of all possible configurations, right? So I'm doing the averaging of all possible configurations of the chain, and then if I rewrite this, I get this, and but what I'm doing is that I'm averaging basically all of these vectors over all possible configurations. But each vector, can point in any possible orientations. If I have one orientation in this direction, this can also point in the opposite direction. So if you add all of the possible configurations, this becomes 0. Okay, and that's not surprising at all, because if this is my first chain and the bottom, okay, I keep doing this. Oh, this is how I lose my DNA damage. Okay, don't smoke. This is what happens. I used to smoke. Okay, so basically, when you let it settle on the surface, then sometimes this end will point in this direction, and something point. So basically, end-to-end vector will, on average, be 0, right? So this has no meaning. It doesn't tell you anything about the size of the polymer. In fact, this is the same as in random walk. So if you start from here, and I have an equal chance of stepping this direction or in that direction, and then if I look at average position, average over many configurations, the position is still 0, right? So that has really it doesn't tell you anything about the extent of diffusion that occurs over that period of time. So what has meaning is this one. So if you measure this, and then you take the square inner product of that itself. So basically, you measure this length squared. And then you average over all possible, here, there should be a square here, all possible configurations. And this becomes so you just open up this product square into these terms, and you notice that this one is simply 1, right? And then you're summing it over from 1 equal n. So what you get there is just n times L squared. Sorry, this is actually product is L squared, because L this is the length of the unit times the unit vector. So this term will give you n times L squared. But the second term here, when you have two random vectors take inner product and then use average over all possible configurations, this again becomes 0. So what you're left with is just this. Okay. So so then the size of the polymer is actually the square root of that. Okay. So let's imagine that if I started with 25 units, and then each one is 1 inch, what is the average size? Well, using this, it'll be 25 square root times 1 inch, or the average will be 5 inches. Okay. So if you have 25-inch long one, you do it many times, the average size you see is about 5 inches. Okay. So that's the idea. Okay. Now, this model that I just described to you is called freely joined chain model, because there's a joint between the two segments is free. Basically, they can take any possible angle between the adjacent chain units. Okay. And in this formalism, this length is called the Kuhn length. Okay. For the double-stranded DNA, approximately each unit can be thought of as 100 nanometers in length, corresponding to about 300 base pairs of DNA. Okay. And so what I mean by this is that, if you want to describe the size of the DNA using some measures using some methods, then you can describe this is pretty reasonably well using this model. And if you use that Kuhn length as a unit length. So this is highly unrealistic. You cannot imagine that DNA is a straight rod, inflexible straight rod up to 300 base pairs, and suddenly the next segment is entirely free to rotate around the first segment. That's unrealistic, but some of the data can be described pretty well with this model if you use that number as a parameter. Now, what happens? So it's really unfortunate that I destroyed this. Okay.
I guess now we appreciate all the hard work your secretary went through.
Oh yes, yes. But she's getting paid the big bucks, so. And I gave her candies. Yeah. Okay, so now if I apply an external force in the direction of stretching the DNA, okay, then what happens is that average is no longer zero. Right, because now there is a direction that is favored, okay? Meaning that each individual segment would like to be aligned along this direction, okay? The minimum energy state for each segment will be actually along this direction. Actually, there's an equivalent system in physics. If you have spin in a lattice, and then if you apply magnetic field, right? Without an external magnetic field, if you ignore interaction between different spins, right, then they are pointing in random directions. There's no net magnetization. But if you apply magnetic field, there's a net magnetization. But if the field is weak, then there's still a lot of random, thermal fluctuations. So net magnetization is not, as bad as it can be. But if you apply really strong magnetic field, everything is fully aligned, then you get basically n times each spin's magnetization, right? Same here. So if you apply force externally, at low forces, they are still fluctuating, but at high enough force and it's fully stretched, then you end-to-end distance, the length of this vector will be that, right? But why do you need to apply force to stretch? Because actually you're fighting against the entropy. Okay? There's nothing actually here each unit is not being stretched. You're actually simplifying the entropy. So this force that you apply, or the resisting force by the DNA against your pulling force, is called the entropic force because of this effect, because it's really purely entropic effect. Okay. So to calculate that, so let's say that this is the z direction, and you're applying force in that direction. Okay. In z direction. Then what you're doing is that you're actually applying external potential, okay? And the external potential that you're applying, I can call it, well, is fine. Let's call it U. This external potential you apply, and it'll be for by z, I mean the projection along the z-axis, okay? z1 and z2 and z3 and so on. So this energy is minimized when z is maximum, right? Along the direction of f. That's what I mean by this. So then this is your energy, and that'll be minus f times n equal 1 to n z n, and that'll be where that is the angle theta. Okay. There is a potential energy that you include. So then, now if you want to calculate, okay, what is the ensemble averaged end-to-end distance along the z direction, then this will be, you know, so what I'm doing here is simply, you know, so z is what you're trying to average, and the rest of it is simply the probability that it'll have certain z value in that direction. So you know, if you want to use the language of partition function, what's in the bottom is the partition function, and so on. Okay. So and then omega 1, omega n, and so on. This is the angular space, and you're averaging over all possible angles here. And if you do that, then in the end, what you're getting is n times L hyperbolic cotangent alpha minus 1 minus 1 of alpha, where alpha is and this function is called the Langevin function. It is precisely the mathematical form that you get if you're trying to calculate the average magnetization as a function of applied magnetic field. And this function will, you know, this z average as a function of force will start 0 at 0 force, end-to-end distance averaged is 0, and then it'll eventually saturate to n times L. So it'll approach that. Same as the magnetization, right? Magnetization as a function of B field will be 0 at the beginning, and then it'll saturate. So you have something like this. The question is that, okay, roughly, what force do you need to apply to get a big effect? Have the change, a large change. That can be, obtained from this scaling factor. So alpha is defined as this. So you get a significant change in the length of this guy, big deviation from the 0 length. If you apply a force such that this becomes about 1. Okay, so this means that you need to apply a force that is about k over t divided by L to actually stretch the DNA substantially. So let me just put in some numbers to make sense of what I just said. So double-strand DNA, the relevant length scale here, Coulomb length is 100 nanometer. That's just based on many different kinds of experiment. And KT can be written as a nanometer times piconewton, right? So that was 4 piconewton nanometer. So then the relevant force scale here will be meaning that even 0.004 piconewton is enough to stretch the DNA substantially. So I've been saying that, well, relevant force scale in molecular scale is about a piconewton, but in this case, it's actually more like a femtonewton. The reason is that the relevant length scale here for double-strand DNA is about 100 nanometers, not a single nanometer. Okay? So this says that applying 40 femtonewton is enough to stretch the DNA substantially. Questions? It turns out that this model, measuring, predicting the length of DNA as a function of applied force, doesn't work. Okay? When people actually measure this experimentally, so I'll tell you about a model that worked after the break. Okay. Did you get some candies?
There's a homework due. I think there's a homework due.
Yeah, you need to collect them. Yeah.
Thank you for the chocolates.
No problem, no problem.
Excellent. Sorry I was late today.
No problem. Yeah, but you missed an interesting video.
Oh.
So Ido told me that he likes this. Yeah.
As much as you want to know.
Huh?
That's good.
Is this how you're actually writing now?
Yeah.
But are you writing such? Oh.
So it's pressure sensitive.
Uh huh.
So
It's just like a Galaxy Note.
yeah. Similar.
Does it? Do you have to worry about your palm?
Oh no. So when
Does it reject palm?
yeah, it does.
Ah.
It can detect the pen.
Uh-huh.
So if you do this either way,
Oh, wow. And the new one has a better battery life, I heard.
oh, I haven't checked it. I think for me the biggest improvement is that the angle is only set at a certain angle, so it's too perpendicular.
I see.
But the new one you can lean back.
I see. And what about the cheaper one? Yeah. Because that's not really good for.
Yeah, so I think the biggest problem is with the hard disk memory size. So after you install your operating system, there's no much more space.
So how much you have? 128?
Yeah.
Here?
128, yeah.
Makes it hard to address. Taking things that you know and applying them to things that was tried before. That becomes harder and harder as you become more and more isolated. Disciplines or sub-disciplines. Yeah, it's true. Deep philosophical subjects.
Ah,
And candy.
candy.
Thank you again for that.
Seems like a good combination.
It's a hard crowd.
All right, so let me resume. Any question about the freely joined chain model?
Freely, what?
Freely jointed.
Oh.
Freely joined, or actually, I'm not sure whether there's a T there or not. Freely, there must be. It must be just freely joined. You know. Okay. So that is the simplest model of a polymer. It's basically it's a random walk. You know. It's a random model, right? So in the random walk model, you take a step of length, you know, certain step size, and after a certain time, and just like the key point here is that the size is proportional to the square root of the unit number. Okay, so that is a key point. And just like in diffusion, if you take 100 steps, then the actual distance you travel will be actually square root of that times each step size, and so on. So for the lambda phase DNA that we discussed at the end of the lecture last time, this has 50,000 base pairs in length. It has genomic DNA of a virus infecting an E. coli cell, 50,000 base pairs long, and approximately 16 micrometers, or 16 in length, fully stretched. Okay. So then L is this, and for double-stranded DNA we said that L is M, therefore n number of Kuhn length units in that length is okay. So in the lambda phase DNA, you have 160 repeats of that paper clip, if you want. Okay. So then, what is the typical size of lambda DNA? That will be square root of n times L. So it will be 160 times 100 nanometers. What is this number?
4 times the square root of 10.
Okay. Okay, so that's about 14.
12 or 13. Oh.
13, right?
Yeah.
Yeah, because 13 squared is 169, right? So that's about 13 times 100. So if you have a lambda phase in it, fully stretched 16 microns long, but if you let it just fluctuate in solution, then it will relax down to a spaghetti coil of this length. So that's the idea.
What's small l?
It is the Kuhn length for double-stranded DNA. Fully joined chain model actually describes the DNA pretty well, except for the force extension curve that I described. So that's what I mean. So how do actually people come up with this kind of information before you were able to stretch the DNA? Well, there's a method called dynamic light scattering, and light scatters more when the object is bigger, and so on. So based on that, you can estimate the size of a polymer when you vary the length of the polymer. And by looking at the scaling, you can deduce actually that Kuhn length. But the measurement is pretty crude, so maybe it's not surprising that a simple model like that still gives you a pretty good agreement. Anyway, so that's the idea, how you can utilize this. This model. All right, so in 1992, I believe, Carlos Bustamante, who is now at Berkeley, his lab actually showed that you can stretch a single lambda DNA molecule using some kind of single molecule manipulation and measure the force applied and measure the extension. So they could measure the length of the DNA as a function of force. And they force and the extension of the DNA, if I use the same term below, and then they got data that kind of look like that. Okay, and as you apply higher force, Z then is eventually saturate. Just like what I do here, right? And then they fit the data using this formula. Sorry. And then their fit was excellent here, and then was not so excellent up there. Okay. So and then later people theorists then came up with a modified version of a different model, and then they showed that model fits the data perfectly. Okay. And it turns out that what I'm doing here is actually log scale. Okay, it fits the data 3 decades. 1,000-fold range in force could be fit extremely well. So that's actually quite surprising. So this is called WLC model, and again, neither fully joined chain model or this model actually assumes anything about the chemical structure of the DNA. Okay, it just considers the DNA as some kind of polymer. Okay, idealized polymer, but the fact that data can be fit so well over so many decades was really quite surprising. Okay, what is WSC model? It actually stands for worm-like chain model. So the idea is the following: you describe the chain not as rigid units connected freely, but you just describe the chain as actually a continuum, like a beam. Okay. But then it has certain flexibility, and the idea is that once you go from here to a certain distance, it still remembers where initial orientation. But if you go far enough, then actually this guy's orientation has no relation whatsoever to the initial orientation. Okay, it's like a worm, moving around. So in this model, what you can do is that, okay, so this is the contour coordinate S along this length, right? So this will be your point S1, and this will be your S2. And then let's call this a 0 and then S, just to simplify. So that along this direction, that's 0 and then S. And then here you can define a tangential vector T1. And here also you can define tangential vector T2. And then let's think about taking a dot product of the two vectors. And then, but once you know the length between the two vectors, you can also have many different kind of polymer configurations, right? So you average over all these configurations. And what will be the functional shape as a function of S? Okay, it's pretty easy to consider some extremes. So this is S. That'll be this function here. When S is 0, two are identical, right? So that's just 1. Okay, so it starts from 1. Then when S is very, very large, then this guy has no relation whatsoever to this vector. So they are independent of each other. So then if you take the dot product of two independent vectors and then average of all possible configurations, that should become 0, right? So you should start from here. It should go to 0. And I claim that it goes like this, exponential decay. Okay? So this actually decays exponentially as a function of S. And then the decay length scale. Okay? This LOP is called the persistence length. So this is really the definition of a worm-like chain model. If you have a chain that satisfies this model, then two tangential vectors separated by distance S along the polymer, and then take the dot product and take the ensemble average, then it follows this exponential decay with a decay length given by this length. Okay? Now, for double-stranded DNA, persistence length is and notice that this length is like half as long as the cone length. That is actually the standard feature of comparing the two models. And intuitively, it kind of makes sense. because according to this model, if you go over 50 nanometers, right? Let's say this is 50 LOP, then you lose some orientation information, but not completely, right? So after going half the cone length, you lose a lot of the information about the orientation, but not completely. So that kind of makes sense, right? And yeah. So if you use this model and then do some approximation, you can get a really beautiful fit to this curve. And again, what's amazing about this is that DNA is a complex chemical molecule. I told you about 5 prime, 3 prime, 1 prime, 2 prime, hydroxyl, 3 prime, phosphate. All these things are there, but you don't even have to know anything about this to explain, this high quality data over such a large range. So this model has been used to describe the DNA properties, and also to explain and to calibrate a lot of the DNA-based experiments. So it has been really, really useful. It also has been used to parameterize mechanical experiments on proteins that are actually already unfolded. So we can discuss this more next time we come back. Okay? Any question? Yes.
I remember either in notes or in homework recently, I thought it was the worm-like chain model that has like a minimum or an angle that you usually find between two DNA. That's what it was. It was in Klaus Schulten's notes. Oh. Isn't there?
Okay, so now you heard it twice. You know that it's important. Okay.
Yeah. But isn't there any of these models that contains like an average angle between two segments?
So yeah, that's a different variant of the freely joined chain model.
Oh.
Yeah.
Yeah, okay, thank you.
Okay, so I'll see you in about 2 weeks. Next week, again, Jingyi Fei, who was there earlier today, will give a lecture on Tuesday on FRET and super resolution imaging. I think it'll be fun. And then she'll show a couple of videos too. And on Thursday, you'll actually visit my lab in the groups of 5. And so one group will do a FRET demo in the lab, and the other will do super res demo. And then in the middle of the class, you'll switch, okay? So you'll do both on Thursday next week. And Jingyi will tell you where to show up, the lecture next week. Okay, thank you.